Hello, everyone, and welcome to today's webinar, De-risk Early and Accelerate Antibody Development with Mass Photometry, Column-Free Aggregation Assessment and In-Solution Binding Mechanisms. I am Cassie Soltman of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Refine. To learn more, visit refine.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. You may also use the Ask a Question box to let us know if you're having any trouble seeing or hearing the presentation. I'd like to now welcome our speaker for today's webinar, Dr. Michael Fiebig, Chief Scientific Officer, Absolute Antibody, a Vector Labs company. Michael, you may now begin your presentation.
Thank you very much, Cassie, for the kind introduction, and thanks to the Refine, Absolute Antibody and LabRoots teams for making this webinar possible. I'm Michael Fiebig, and I'm the CSO of Absolute Antibody, a Vector Labs company, and today I want to tell you about the work we've been doing together with Refine to apply mass photometry to some of the more challenging antibody constructs we work with. Though let me first tell you a little bit about Absolute Antibody. We're an antibody reagent and service provider exclusively focused on recombinant antibody technology, founded with a vision that everyone across biomedical research should have access to recombinant and engineered antibodies, not just researchers in pharma. As part of that, we offer an ever-growing catalogue of recombinant antibodies, as well as a portfolio of recombinant antibody services, including antibody sequencing, engineering, transient expression, and subsequent purification. We were founded in 2012 in the UK, and you can see our production site at the Wilton Centre in the typical northeast England sunshine on the right. And in the summer of 2024, we merged with Vector Laboratories, further expanding our joint service offering into specialty chemicals and bioconjugation services, as highlighted on the slide with a nod to our biodesign toolbox. This slide really sums up the core of what we do at Absolute Antibody. Once you have the sequences of an antibody and you know the sequences of the variable domains mediating antigen binding, you can convert this antibody into any format to suit your specific experimental needs. These can be naturally occurring antibody isotypes as shown in the first three rows, or artificial designs such as recombinant fragments or multispecific antibodies. Indeed, multispecific antibodies have been keeping us quite busy over the past 10 years, as these are very promising therapeutic modalities. On this slide, of which you can find an interactive version on our website, we've tried to summarize some of the weird and wonderful constructs we've made over the years. And I'm sure you can appreciate that as we make these constructs, we can sometimes get unpredictable behavior of the final molecules, which gives us interesting analytical challenges. Initially, purity tends to be the biggest concern, and this can be purity of the protein itself, but also purity of the correctly folded species. A big part of that is aggregation, but also degradation, of course, which directly impacts manufacturability, as you will lose precious material to that, but also the impact of that on downstream immunogenicity and stability if you're planning to develop a therapeutic. Likewise, activity is a big question, especially if you're building something multispecific where you need to differentiate between binding to each target individually, as well as the impact of the architecture of the antibody on its binding activity. So in the following, I want to walk through two case studies we performed with Refine and their mass photometry platform. The first case study we're going to look at is a comparison of mass photometry with size exclusion HPLC, talking to those first analytical challenges I'd outlined. In the second case study, we're going to expand that into bispecific antibodies and look at activity of those, talking more to the latter analytical challenges I'd mentioned. So here goes, mass photometry versus SEC-HPLC for aggregation assessment. The benchmark method for assessing aggregation states of protein is size exclusion HPLC, where proteins are separated on a column containing a resin with porous particles. When passed through this resin, smaller molecules are able to enter these porous particles more readily, whilst larger molecules flow through more readily, leading to separation by size. These separated species can then be detected by a UV detector and absorbance plotted against retention time. The total absorbance observed is proportional to the extinction coefficient, and thereby to a degree to the size of the protein. But with a bit of data trickery, one can correct for this to get quantitative abundance data. Size information can be obtained by comparison to standards, though resins may have a limited linear window of resolution. A typical SEC-HPLC trace we would see for, say, an IgG looks like this, with a main monomer peak and a little bit of a high molecular weight peak from antibody dimer. Now, mass photometry is a light scattering based technique that measures the interference between light scattered by molecules near a slight surface and light reflected from the surface. The intensity of the signal is linearly proportional to the mass of the molecule. So as these molecules, which are in solution, are observed by the detector, you can get information about their mass and actual counts of these molecules. And this linear resolution is quite important to note, as this is a huge differentiator to SEC-HPLC, where a specific resin only gives you a limited linear window of resolution. And this is what it looked like when Nina, who's going to be around for the Q&A later, and Gogs from Refine came over to our site and set up the mass photometer next to our SEC-HPLC to run the experiment, which we're going to be showing you the data of. This is in real time. It's a simple question of pipetting a few nanograms of material onto a chip, closing the lid, and then watching the different particles with different scattering intensities appear in front of the camera. It took minimal calibration and we were able to resolve different IgG assemblies pretty quickly. The runs only take a minute, and so we're done with the mass photometry part of the experiment by lunchtime, which I thought was quite handy. This is the kind of data set we will be looking at in this study. We have an SDS page gel on the left under non-reducing and reducing conditions, showing intact antibody bands and bands corresponding to heavy and light chains respectively. Then we have a SEC-HPLC trace and the mass photometry readout converted into a similar format for comparison purposes. On some slides we'll also have a data table. In an ideal world, the readout from SEC-HPLC and mass photometry should match up, as they do here. And indeed, we were able to run lots of well-behaved antibodies and got excellent agreement. However, while these scenarios are nice, they're a bit boring, right? So let's look at some more interesting samples. Here we have a human IgG1, which by SEC-HPLC shows a main peak corresponding to the monomer, maybe with a little tail, and a peak containing dimer with potentially a shoulder for trimer. It's difficult to see. Even if we were to stretch the image, the peaks are just overlapping. When we look at this by mass photometry, we get much better resolution and can see some fragments, as well as get clear counts for the dimer and trimer species. Between the two methods, the percentages for the dimers and trimers are quite different, but if we factor in that SEC-HPLC measures absorbance, and even just crudely correct for the fact that the complexes are 2 or 3 times as big, then we actually get pretty good agreement in those high molecular weight species. The resolution of SEC-HPLC across a wide range of molecular weights becomes even more of an issue when you're looking at larger assemblies like IgMs, in this case a hexameric IgM, that stretches all the way to over 1000 kD. By SEC-HPLC, you get a single, main high molecular weight peak with some fragments. When we ran this on mass photometry, at first we got very little high molecular weight material, which was a bit concerning at first. Though the team at Refine suggested using their mass fluidics rapid dilution chip, which essentially allows measurement of complexes before dilution effects have had a chance to fully manifest themselves. And lo and behold, we see a much more pronounced ladder, still a ladder, of high molecular weight species that you wouldn't have any idea being present on just SEC-HPLC. And we can replicate a similar story with another slightly problematic IgM here. Next, we looked at a scenario of a seemingly homogeneous antibody. Seemingly. The retention time of this antibody was shifted from what ought to be 7.5 minutes to over 9 minutes. This would indicate a much smaller molecular weight, though we see no indication of that on STS page. Well, the non-reduced band is running a little low, but not that low. Looking at it by mass photometry, we got textbook antibody data at 145 to 150 kD. With just SEC-HPLC, we might have been concerned about the in-solution state of the antibody, but this was clearly an artifact. Now, there are lots of reasons that can cause this, often due to column or pressure system damage, but we could run other antibodies afterwards on the same system with no issues, and the second we ran this antibody, or even a different lot of this antibody, we saw the same thing again. There's clearly something interacting funny. We've seen the good, we've seen the bad, now we're going to look at the ugly. This is a mouse IgG2A that looked completely normal on STS page, but try as we might, we couldn't get any signal at all on SEC-HPLC. The peak that we're showing is at such a low absolute scale that basically makes this background noise. We of course repeated this, and the protein is consistently there by STS page and consistently not there by SEC-HPLC. When we turned to mass photometry, it was right there where we expected it to be. Clearly, SEC-HPLC has some artifacts that we might have been able to fix with some further optimization, but if, like you're running hundreds of different antibodies in different formats through your QC platform every month, you just don't have time to optimize for every single construct. And that wasn't the only example. We have a similar story with a human IgG1 here. And a final example of a failed SEC-HPLC experiment, this time on a Fab fragment. Here you can even see a fairly accurate representation of, let's call it a high molecular weight smear in mass photometry mirroring the data from STS page. I'd like to conclude this part by pulling up a little comparison of mass photometry and size exclusion chromatography. You'll be able to read through these faster than I can walk you through them, but I think the key points around separation method, or lack thereof, and resolution, which have been key factors in the artifacts we just went through, are of particular note. Likewise, sample requirements and runtime give mass photometry a bit of an edge over the industry standard SEC-HPLC. Now, these kinds of artifacts in SEC-HPLC that we're showing are, of course, very rare. However, for us as a protein engineering and expression service provider, when they come about, it's always inconvenient. We use SEC-HPLC data during the purification process to track the in-solution behavior of a molecule and to assess whether or not additional purification steps might be necessary. Likewise, as a final QC package, it's incredibly important to our clients to know what their molecules look like and how they're behaving. Do they have a propensity for aggregation? It's a key deliverable we provide. For some clients, at least initially, it's even more important than the protein itself. So in order to allow bad molecules to fail early, we're reliant on robust analytics. And if need be, we can then start fixing those molecules. Now to a slightly different application of this. As I had alluded to earlier, we do a lot of work with bispecific and generally multi-specific antibodies. A while back, we performed a study where we took the anti-HER2 antibody trastuzumab and the anti-CD3 antibody OKT3 and created a whole panel of different bispecific antibody constructs. We've been using these as a reference panel for a wide range of different applications, and so we thought, hey, let's take a look and see what we can do with these in a mass photometry setup. First off, we looked at the parental control antibodies and various bispecific antibody designs individually. We got pretty nice data corresponding to the predicted molecular weights along with some small amounts of aggregates in these. The next step was to add antigen and see if we can see the complexes that form. We can see that in this titration experiment, we're moving from just antibody in the absence of HER2 antigen to initially forming a complex of antibody with one HER2 moiety to a complex of two HER2 per antibody, and eventually plateauing out at the full complex and excess-free HER2. I think it's pretty neat that we can see the single-bound and double-bound complexes in there. This is a lot more difficult in other binding analysis methods. And if you're now reminded of biophysics lectures you might have taken during your undergraduate, where you had to calculate binding constants from data like this, well, that's exactly what our friends at Refine did. So this is quite a big piece of work. working out ratios of antibody and antibody HER2 complexes and calculating KDs from these. But the KDs fall in the range of what's been reported for trastuzumab. There's probably a little bit more that could be tinkered with the conditions to get to grips with absolute values, but we can clearly see the expected complexes and that we're in absolutely the right neighborhood for affinity constants. And then we can get those not just for a single binding event, but tease them apart for multiple binding events. Doing the same for CD3 binding was a lot more difficult, as we'd used a humanized OKT3 variant that has very low affinity. This is quite intentional to avoid binding in the periphery and rather make use of avidity to trigger T-cell activation when the bispecifics are bound on a tumor cell. So in the same setup used for HER2, we saw no complex formation. We then went back to using that mass fluidics system we'd already seen in the context of IgMs that allows us to observe complexes found at a high concentration before dilution effects could set in. Here we clearly saw the complex of antibody and CD3 antigen. This could also be replicated with some gentle protein cross-linking, which very clearly fixed the complex. With the cross-linking trick in hand, a similar data set across the bispecifics with CD3 could be gathered. In blue, you have antibody and antigen just mixed, and in yellow, with cross-linker. You can't get affinity data from this as it's not an equilibrium observation, but it's great to see all the expected complexes forming. Well, almost all. If you look at bispecific A in the top middle graph, you might think that the second scFv of this Morrison format antibody isn't binding. Well, if we were to zoom in really closely, there are actually some of those complexes that were detected, just very few. This is either just a function of the little complex being formed in the first place and low chance of a second one forming accordingly, or there is some steric hindrance further complicating antigen binding. These data will be coming out in a white paper shortly, so if that's of interest to you, make sure we have your details to send you a copy. And of course, the bispecifics dataset wouldn't be complete if we didn't have an example of engagement of both HER2 and CD3. The top graph showing the antibody HER2 complex forming in solution, and in the bottom one, the CD3 antigen joining the complex with both HER2 and CD3 bound by this bispecific. I think it's absolutely great to be able to see our bispecific molecules in action like this. To conclude, I hope I've been able to convey some of our excitement around these data, how they can not only help us look at our antibodies with fewer artifacts, but also how we can confirm structure-dependent activity of complex molecules. We always draw pictures, or more often make PowerPoint slides around what these molecules do. Seeing these complexes actually form is pretty amazing. And I look forward to seeing how this story develops further, especially as we start delving into things like proteins pegylated with Vector Labs' DPEG technology or antibody drug conjugates as part of our biodesigner toolbox. Naturally, this work wouldn't have come together without the teams at both ABAB and Refine. I'd particularly like to thank Holly, Lexi, Evie and Stephen from ABAB's QC team for acquiring all the SEC-HPLC data, as well as Nina and Gogs from Refine for making the trip up to Wilton to run the mass photometry experiment right there alongside us. I'd also like to thank Patty from ABAB and Sal Mansour from Refine for helping put these slides together and organize the webinar. And of course, thank you all for spending time with us today, and I'd be happy to answer any questions you might have.
Thank you for your informative presentation. We will now start the live Q&A portion of the webinar. Joining us for the Q&A session is Nina, Senior Scientist at Refine. If you have a question you'd like to ask, please do so now. Just type your question into the Ask a Question box and click Submit. We'll answer as many of your questions as we have time for. Let's go ahead and get started. Our first question, it looks like, is for Nina. This question is asking, could you comment a bit more on the difference in quantifiability of data obtained from MP versus SEC-HPLC?
Yes, thank you. That's a great question, and we got this question asked quite a lot. So one thing that it's really important to know is that when we talk about SEC, these measurements are mass weighted, and when we talk about mass photometry, these measurements are concentration weighted. Now, what does that mean? When you have your protein in solution, your protein will absorb a certain amount of light. This depends on the excitation coefficient, which is property of every protein, and it depends on the amount of aromatic amino acids you have in that protein, mainly tryptophan. So let's say you have a monomer, and that will absorb a certain amount of the UV light. But if you have dimer of the same molecule, you will have twice as aromatic residues, and therefore you will have twice of absorption of UV light, which might look like you have twice of dimer than monomer. This is what we mean when we say that this is mass weighted, where heavier species will absorb proportionally more. On the other hand, mass photometry relies on a single molecule counting, and the outputs of mass photometry are histograms, where each species presented has been counted once, independent of its size. So again, if you have a scenario where you have monomer and dimer, you will actually count one monomer of its size and then dimer, one molecule of dimer, which has double the size. And as Mike already said, you can tweak this a bit, and you can transform your SEC measurements, which are mass weighted, to concentration weighted, and you can do it the other way around. We actually did that with our MP data, and not surprisingly, the data were almost identical in terms of quantification as the SEC data.
Nice.
Excellent. Thank you, Nina. All right, moving on here. Our next question is for you, Michael. This question is asking, can you comment on the mass range of mass photometry compared to SEC?
So in terms of resolution of the masses and sizes of the molecules, yeah, sure. So mass photometry. In mass photometry, we sort of have a range of that kind of starts at around 30 kilodaltons and goes up to 5 megadaltons. I know that there's some development in expanding that further, also into the lower molecular weights, so down even further from 30 kilodaltons, because there are of course interesting proteins below that. But that is still a little bit difficult. For SEC-HPLC, it really depends on the kind of resin you use in your column. Now, the standard resin that we use resolves quite nicely, sort of in that range of 10 to about 200 kilodaltons. Now, around the 10 kilodalton mark, so 12 and a half kilodaltons, that's where you have, say, your nanobodies. And then 25 kD, you get to single chain FVs, and sort of down there, you do have some quite topical molecules for the kind of antibody space that we're working in. And then you go up and have you have your normal antibody 150 kD, and once you start bolting things onto that, you quite quickly get to 200 kD. Above that, though, yes, you start squishing things together a little bit, and this is where the extended linearity of mass photometry does come with an advantage to these kinds of analyses. Yeah. So, long story short, 30 kD to about 5 megadaltons, and for the SEC, at least in the setup that we use, about 10 kD to 200 kD.
Okay, wonderful. Thank you. Okay, our next question here that we have is asking, what are the industry perspectives on adopting mass photometry as an orthogonal method to SEC and antibody characterization?
Yeah, that's a very good question. Well, as you've seen in the data we presented, and I think as also mentioned in the talk, for a lot of molecules, you're kind of used to seeing SEC-HPLC data, and when it's well behaved, that's what you want to see, and I don't think you will bat an eye at just seeing that. That's when you start seeing slightly unusual things. That's when the need for an additional method comes in, and that's also where we've received quite really positive feedback on when we've been able to present that, especially in some of those cases where we had slightly funky SEC-HPLC data. I have spoken also with other service providers, for example, a service provider that specializes in generating crystal structures and cryo-EM structures. They use mass photometry a lot as a really good measure to identify whether or not there are any impurities in their sample. And they say it's much far superior in that application than any other methods they've had. So I think there's definitely a growing sentiment that the advantages it offers are worthwhile. Though whenever you're trying to align or potentially in some aspects displace a well-established method like SEC-HPLC, it's of course an uphill battle. But I think, as we've seen today, there are lots of advantages of why you would use this, and especially if something like the lower limits can be improved upon, you'd be hard pressed to argue against mass photometry, I think.
Okay, wonderful, thank you.
I don't know, Nina, maybe something from your side. I mean, you're obviously talking a lot more with the kinds of people directly talking about the mass photometry. Is there something you would want to add to that?
No, I think you summarized it perfectly.
Sorry, I didn't want to throw you under the bus there.
Okay. All right, we have a lot of questions coming in. So for you watching, please continue submitting your questions. These are excellent ones. Our next one that we have is asking, I believe this is to you, Michael, is the mass photometry method compatible with high throughput screening in early stage antibody discovery?
For that, if you really wanted to do it in a high throughput screening setup, I think you would want to have some sort of automated sampling setup for that. We do. Dina, does it exist? So I think I'm going to have to pass this question on.
It does. We have auto 2MP systems, which are automatic, and you can analyze up to 26 samples. So you can just easily set up your, let's say, purified last step purification antibodies. You can set your experiments up and just walk away, and your data will be analyzed. Your antibodies will be analyzed with a high throughput auto and 2MP system. So yeah, we have that.
And I think especially in something like this, for these screening purposes, it's excellent if you can very quickly get to grips to what kind of complexes you're forming, if you are forming any complexes, and quickly weed out the kinds of molecules that you just don't want to be working with, if you have alternatives.
That's true, yeah.
Okay, wonderful. Our next question that we have here is asking, can you analyze antibody drug conjugates with mass photometry too?
So that is something we are looking to work on, but with antibody drug conjugates, you often have a lot of them can be a bit hydrophobic, and so if you're working with them in a separation matrix, you can get probably even more artifacts from this than we were showing with just antibodies. But because there's no separation of the molecules in mass photometry, they're just in solution and analyzed as they're diffusing about and coming close to that slide surface. I don't think there should be any reason for why this wouldn't be possible. But we hope to have some data to show you on this soon. And correct me if I'm wrong, Nina, the same also applies to even just fluorescent markers, right? You don't get interference of fluorescent markers on proteins if you did a fluorescent conjugate with the scattering, right?
No. That doesn't happen. Our laser is too strong, so it just the scattered light, the signal, the ratiometric contrast just is the main signal and output you get. But speaking of antibody drug conjugates, just to continue and add my 2 cents on what Mike said, due to the slightly bit complicated properties of the antibody drug conjugates, higher hydrophobicity. So MP can really help, analyze samples in terms of how stable they are they more prone to degradation. You can do experiments in force degradation and stability testing. So that is all very possible with mass photometry. In terms of drug conjugates, that is still below the resolution of mass photometry, which is 30 kilodalton for the time being. So that cannot be at this moment determined by mass photometry, but every other property of antibody drug conjugate can.
Okay, wonderful. Thank you both. All right, moving right along here, our next question is asking, is target binding also label free?
Yes.
So you go
yes.
for it, Nina. You ran the experiment. You go for it.
So the beauty of mass photometry is not only this is a single molecule technique, it's label free. So you absolutely don't need to label any of your samples in order to detect them if they are in range of mass photometry.
Okay, anything you want to add? No? Excellent. All right. Rolling through. Our next one here is asking, does mass photometry, is it more specific and sensitive compared to chromatographic technologies?
Well, we've definitely seen fewer artifacts arising now and we do see, I think this is the example I was showing with the IgM molecules, where once we're outside of the good dynamic range of the column resin that we're using, so what's really creating the separation. At that point, we were getting several species just squished together under one peak. And for those, you actually couldn't tell things apart, because even if there was a little shoulder, was it a peak? Was it what, how much is really under those peaks? And this kind of thing, you get much better resolution on mass photometry, because for each particle you get an individual count and not just a rise and a fall of a UV absorbance as it comes out of the column. And you are seeing individual molecules. So in terms of sensitivity and being able to see individual molecules, I'm not sure you can get much more sensitive than that, really. In terms of concentration, perhaps, Nina, is there like what's sort of the bottom concentration optimum that you might want to put on a chip like that?
For the mass photometry, the beauty of mass photometry is basically that you really need a low amount of samples, so you can save your sample big time. We suggest a range of concentration from 100 picomolar to 100 nanomolar, with optimal range between 5 and 20 nanomolar. Now, if you doubt that your sample might be affected by those low concentrations, like we've seen in example of IgM, when you dilute it, you will lose the bigger aggregates, then there is a possibility of using our mass fluidics system with a rapid dilution, where basically dilution happens so fast that you can still capture majority of the higher molecular species, that is aggregates. But in terms of, if I can just add, in terms of chromatography, it depends. You have, Mike, correct me if I'm wrong, whole range of the designs and the dimensions of your columns, which then have different properties in terms of resolution, sample volume, concentration, etc., etc. But for mass photometry, so the limit for 2MP is 30 kilodaltons to up to basically 6 megadaltons. So anything in that range you can measure, but important thing is resolution. So mass photometry has a really high resolution, which is for a molecule which is around 66 kilodaltons, the resolution is plus minus 25 kilodaltons. So you can have a mixture of, let's say, 40 kilodaltons, 66 and 90, and then you will be able to resolve all of those three molecules, those three species in one experiment, which is really helpful.
Excellent. Thank you both. All right. Our next question here is asked, it says, you've shown how simple it is to take a measurement, but what about the analysis? How difficult is it to use the analysis software for MP?
I think that one I'm going to pass over to Nina as well, because it looked fairly simple to me, but you were all driving it, and I never had to learn the software itself, but it looked fairly intuitive to me. But maybe you take that one.
Software is super easy to use. Basically, as you could see from that movie in the presentation, you load your sample, you close the instrument, you measure your movies, default 60 seconds, and then immediately you can analyze those data in our software DMP, which is for analysis, and AMP is for acquisition. And when you apply a proper reference, which we also provide, is the standard for calibration, you can literally within 1 minute, you can analyze your sample and see what species you have in that sample you just measured. So it's really, really straightforward, very intuitive, and very simple to use.
Excellent. Thank you, Nina. All right. Our next question here that we have is asking, do you see antibody aggregates binding to antigen by mass photometry?
Okay, the samples we had weren't overly aggregated, but at least the ones we did the antigen binding of, but we did see antibody dimer binding to one copy of antigen, if I'm not mistaken. So forming IgG or IgG-like dimers isn't unusual for antibodies to behave, and so it still leaves activity intact. So that is something we could observe. Whether or not, if we really aggregated a sample, maybe by mistreating it deliberately, we could still see that. I don't know. Might be an interesting one to do.
We have also, speaking of that, we have module for the antibody aggregation, basically, where you can automatically analyze your antibody samples in terms of stability and aggregation. So that's also very useful for that matter.
Okay, excellent. Thank you. All right. Next question that we have here is asking, do you need a different setup when measuring IgMs as opposed to similar or smaller proteins like IgGs?
No, absolutely. The setup is same. As I said, the window for measuring your sample is from 30 kilodaltons to 6 megadaltons. So if it falls within the range, you can measure it. No special preparation or anything, just dilution of your sample to nanomolar concentration and 1 minute measure and 1 minute analysis, and that's it.
Okay, excellent. Thank you. All right, next question. Can mass photometry distinguish between different classes or isotypes of antibodies, e.g. IgG, IgA, or IgM?
Yeah, I think that question falls in a similar vein as to the previous one. Yes, you will see you will definitely see differences between those subclasses, because you get a very, very precise determination of the mass that you've got. And especially then, if you're thinking about IgA forming dimers, IgA with J chain, you can get a very, very good characterization of the sample that you have with something like this. And yeah, an IgM again, there it will look very, very different to an IgG sample. So for something like this, it should be, it's very suitable.
Okay, wonderful. And it looks like we have one more question here today. This last one is asking, can mass photometry be used to study post-translational modifications, e.g. glycosylation of antibodies?
So we are those post-translation modifications are adding rather small amount of mass to the proteins. So if you want to, for example, analyze if your protein has, one glycosylation, N-link glycosylation, and the other protein doesn't, you won't be able to see that. But what mass photometry also is good at is we can, due to the properties of heavily glycosylated proteins, which can be sometimes tricky to work with, mass photometry absolutely has no issues with measuring those samples. And if you have a heavily glycosylated protein, you can roughly estimate how much that adds to a mass, and then you can see if you have glycosylation failed or working fine in your mammalian expression system. Then you will be able to distinguish. Definitely, for example, if you express protein in bacterial cell, heavily glycosylated protein in bacterial cell where there's no glycosylation, and in mammalian cell, and you put those two onto MP at the same time, you would definitely be able to resolve that.
Okay, wonderful. And we had one more pop in, so this may be a follow-up. This question's asking, can mass photometry distinguish between normal and abnormal antibodies based on molecular mass?
Well, it depends what the nature of the abnormality is. If it is something like, coming back to the glycosylation patterns, if it is increased fucosylation, for example, then I think the size difference that might be in part, the mass difference might be very subtle. If it's something around different, let's say, different hinge formations, so some antibody subtypes can rearrange their hinge a little bit and form slightly funky structures like that. There, the actual mass isn't changed, so that would probably be a little bit tricky. However, I think with both something like post-translational modifications or things like abnormal folding, if you have probes, let's say nanobodies or small lectins that can selectively bind to these abnormal structures, and if you were to co-incubate them with antibodies, you would see antibodies that are normal or and haven't bound to anything, and then you would see any abnormal ones that might have bound to another protein as a sort of label, because that will increase the mass substantially. And so you can still then get quantification of normal and abnormal, or modified and not modified, by using a specific probe or protein label afterwards to induce such a mass difference. So that could be an idea if such tools exist for the particular modification or in question. So if you can make it more heavy, it'll work quite nice.
Excellent.
Good question. I like that.
Perfect. I want to thank you both for your time today, and Michael for your important research. We would also like to thank Labrits and our sponsor Refine for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today, and those submitted during the on-demand period, will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. Labrits will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.
Thank you all. Bye-bye.